today I start with the second part, or I finished with the second part. If you remember yesterday, we stopped at this stage. So I try to show you that at the molecular level you can see a very high level of variability. It's a stochastic variability. The cell, as a result, the cell also displays some kind of very highly variable behavior. And we also saw that to reduce this variability is only possible with a very high energy demand, and the cell simply cannot afford that cost of energetic cost to reduce all cellular processes. So the question now that we know that the cell functions in a more or less orderly way, so how to reconcile what we observe at the cellular level as an ordered functioning with this very high stochastic behavior at the molecular level. Let's take it from a completely theoretical point of view. Usually we frame the problem using a very simple postulate or assumption. This is this, oops, we have to come back. So this is the stability. So we suppose that the cell is stable, that the living matter is stable and functions in a regulated and stable way. And if we observe variation, this is because there is some kind of noise. So the starting point is stability, the postulate is stability, and we build all the rests on this postulate. From this viewpoint, variation is a noise. Noise means something which is bad because it destroys order. So from this point of view, variation is something which must be reduced to go back to the more orderly functioning as possible. So most of the, or at least a large part of the studies on variability uses this postulate. I'll show you some examples. So you can find a lot of papers studying variability from this standpoint. They say, okay, that living cell does things in an orderly way, and there is some noise, and in some cases this noise is reduced, in some cases this noise can be even useful. We have examples already discussed this week, or at least we touched this question this week. For example, at the populational level, cellular variability can be useful for the survival of the population. So it can be an evolutionary favored strategy for survival. Also, other studies about the stochasticity in cell differentiation also show that in some cases stochasticity is something very useful. A particular example of this is the antibody production in lymphocytes, in B lymphocytes or T receptors in T lymphocytes, where you have a stochastic rearrangement of pieces of DNA and producing an antibody which at the end will help the whole organism to survive. So these are all viewpoints based on the idea that everything is ordered and there is some noise around. This is a paper Vidya sent me at the beginning of this meeting, published by our first speaker, Stuart Newman. He's discussing this exactly from this standpoint. So I took this picture because it illustrates well the basic idea. You have something which is completely deterministic at the beginning, and we add noise at each level. At the end, we get some heterogeneity, which is useful because it participates to the cell differentiation. So now what I propose, but it's certainly not sufficient to explain everything. We saw that many of this white noise can be reduced by statistical averaging in space and time, but it is clearly not sufficient. So I propose to take this problem other way around. So instead of considering the ground state of the living matter is stable, we can inverse and we can postulate variation. We can say that everything is variable all the time. Variation is the normal state of the living matter. So what we have to explain in this case, we observe stability because it functions in an orderly way over time. So we can find explanation for the stability and not for the variability. So let's put other side around. So let's try to see if it works. We can take as example, for example, the gene expression. We saw the gene expression is highly variable and it's mostly stochastic in individual cells. At the same time, we can have ordered behavior in the cell. The first thing we already touched yesterday and I mentioned is the spatiotemporal averaging. I'll just show you two examples, published examples. But before, yes, what is the basis of this spatiotemporal averaging? We can quote Kolmogorov, who said that all epistemological value of the theory of probability is based on this. The large-scale random phenomena, their collective action creates strict non-random regularity. So this is the basic idea of everything which can be considered as spatiotemporal averaging of stochastic changes. And we can also quote our friend Schrödinger. In the same book, he explained very clearly this idea, and it is still true, especially for biologists. Non-physicists find it hard to believe that really the ordinary laws of physics, which he regards as a prototype of inviolable precision, should be based on the statistical tendency of the matter to go over into disorder. So this is something which is really, for a conventional thinking, it's very difficult to accept, but this is the case. So let's see those examples. Excuse me. We talked about the Drosophila patterning and the French flag model and morphogene gradients along the embryo, which establishes a kind of positional information for the cell and drives the differentiation and segmentation of the embryo. This is usually regarded as a highly regulated and precise mechanism. And in fact, we saw that the simple diffusion from one end toward the other end can establish a morphogene gradient. And then if you look at, you zoom in the border, because interesting things always happen at position, what you can see is that at the level of the cell nuclei, you see a totally stochastic pattern of expression. What we have here, as they say in the paper, the developmental patterns are not just the consequence of reproducible transcriptional dynamics in individual nuclei, but are the results of an averaging expression over space and time. So this is exactly what Kolmogorov said and Schrödinger said, and what we propose to apply to these cases. So we can take many examples of apparently deterministic mechanisms that determine embryo part tissue formation or pattern formation, and we always will find something similar. So if we go on with this example, this is another paper. They look at the transcript numbers in the cells, the same, the borderline, and they come to the same conclusion. Stochastic expression results in precise and nearly uniform transcript accumulation achieved by straightforward spatiotemporal patterning. So we have many other examples of this type. I go show you this because we mentioned the embryo patterning issue here, and this is also a very classical example, one of the very classical examples in embryology. Also, we see here stochastic expression, gene expression in action. But we know that this is not sufficient. We know this is not sufficient because basically spatiotemporal pattern averaging is based on more energy investment. You need a lot of cells, for example, so it costs a lot of energy for the cell. So there are also other tricks the cell can use to overcome the problem of stochastic variation. What kind of things these are? Now we jump back to have an historical glimpse on some ideas in embryology and genetics. And we have to cite this name, Conrad Waddington, because he is really a determining person in biology. Conrad Waddington was a geneticist and an embryologist, so he bridged these two different disciplines, appeared in the 19th century, and I mentioned that epigenesis was maybe the ancestor of embryology, and this is a science of change and development. And the other science was genetics. Genetics is the science of stability and heredity. And so there's a contradiction between the two. So what Waddington proposed is try to make a synthesis of these two. How to synthesize these 2 points of view and create something coherent and structured, a kind of structured theory about cell differentiation and embryo development. And he said, okay, let's fuse. So he created a new term, epigenetics. Waddington is the inventor of the term epigenetics. Now you find this term in every second publication in biology, usually used in a different context, by the way, but still interesting to see that this comes from the middle of the 20th century. For him, the epigenetics is a branch of biology which studies the causal interactions between genes and their products, which bring the phenotype into being. So this is a very general statement, how he imagined all this. Let's have a look. The most famous way to illustrate his idea is his own drawing. So this is a metaphor, a visual metaphor he proposed. Here what you can see is its individual cell on the way of differentiation in the embryo. And he made this landscape we call epigenetic landscape now. And his idea was that the shape of this landscape is defined by the genome, by the genes. So what happens here that you have a defined surface, genetically defined surface, but the exact fate of an individual cell is not determined by the genes. What the cell does is just to rolling down this landscape, and each time it comes to a bifurcation, there's a choice to do, left or right. Nothing in the cell itself intrinsically defines whether the cell has to go left or right. It is a question of circumstances at the moment when the cell comes here. So small local perturbations, intrinsic perturbations of the cell can push left or right and choose with a certain probability to go in this way or the other way. And then, of course, there are many other bifurcations before coming to the final phenotype, and each time the choice is done basically by chance. However, the shape of the landscape will define what is the probability to go left or right. So if you have a lot of cells and not one, because in the embryo you have always a lot of cells, the statistical outcome will be predictable. So this is an idea which is not that original in the statistical way. If you think about Galton, for example, he did exactly the same. But in the context of biology, this analogy or this metaphor can help to bring together genetic determinism and stochasticity and everything which can come within the same framework. So how it works? I'll show you this. This is a simulation taken from this paper. You see here the starting point, and there are only two possible outcomes, and you can already guess what will happen. So the cells under the spontaneous movement come out from this stable state, and they roll down the surface, and with some probability they will go left or right. Some of these points are colored, so you can see that if you follow them, it is impossible to predict from the beginning where they will be where exactly they will end, left or right. So here we have 2 points on one and 2 points in the other, but they all, the pathway was completely random. Of course, this is just a simple example, and there is absolutely nothing to orient the cells in between. What happens if the structure of the surface is a little bit more complicated? What you can see here that the shape of the surface can orient the cells in one way or another, but in any case, the end point will be left or right. This is a completely regular surface, but you can imagine that if this choice is not 50-50 at each bifurcation, then you can, you can have a distribution which will be different. So with a little imagination, you can imagine hundreds of different outcomes, possible outcomes, and this will make, make your organism much more complex, of course. So this is the basic idea of the epigenetic landscape in the way Waddington imagined it, and but there is also another aspect, and it is also very important, but usually we forget about it. This is the original, but this is also a drawing coming from the same book. He represented here the epigenetic landscape view from the bottom. So what we see here, these are the genes, and the lines connecting the genes to the surface show how the genome determines the surface, the shape of the surface. And as you can see that there is no one-to-one correspondence between the genes and the points in the surface. Each gene interacts with the other genes. To my knowledge, this is the first representation of a genetic network. He didn't use this term, genetic network, but clearly the idea is there. So this means that the outcome, the surface, the shape of the surface is an outcome of the whole genetic network and not of an individual gene. So the network idea can be developed further. I think that somebody mentioned the first week the name of Stuart Kaufman. He was the first to suggest that if you have a network, that network may have interacting elements. That network may have different or several different stable states. So the idea is developed here. So this is a very simple genetic network. We have already seen this. Only 2 genes, and if they interact with each other, they can create a bistability. So this network has already 2 stable states that can be represented in a 2-dimensional phase plane, but also using a Waddingtonian type of metaphor with 2 wells, 2 communicating wells. Now if you go further and you imagine a much more complicated network with different interactions, then this network may have many stable states, and you can describe this also with mathematical tools, and you can also translate this into a Waddingtonian type of representation. Here, actually, what you can see is that the different stable states are represented as attractors. So these attractors correspond to the fixed cell phenotypes, cell states, and the transition between the attractors can be done simply by coming out from one and going down to the other. Clearly, the transition between cell states can be initiated only by noise, by variation, by stochastic variation. This is a 3-dimensional landscape, but you can add as many dimensions as you have genes in your network. So this idea is developed by Sui Huang, who is a who is a co-worker who worked with Stuart Kaufman. This type of representation is becoming more and more popular. So I have a whole selection of different images from different papers, how they show different epigenetic landscapes and explain cell differentiation using this. This is again coming from a paper by Sui Huang. So we're explaining the same idea that you have a linear genome, networking interaction between the genes. This network may have several different stable positions, and that you can represent it in a state space. Let's say you can reduce it to 3 dimensions, and then you see different outcomes, different differentiated cell types as reaching their attractors after a more or less randomized pathway. Let's try to see if this can work, because this is very nice. This is the theory. The basic idea here is that the interactions within the network act on stochastic variation like a kind of system of constraints which will canalize the cell from one phenotype to another. In this pathway, of course, there's a big part of the chance where each time there's a choice to make, the cell can go one pathway to another. So these constraints are defined by the genome, according to. But if we try to make this idea of networking and constraints within networks larger, we can see that the same idea can be applied at different levels. So let's take a few examples. You remember that I showed you this experiment from our lab using these 2 transgenes inserted in the genome of the cells in 1 copy each, and then examining clonal cell populations, which were very diverse. And we also saw that the amount of the fluorescent protein in the cell varied very quickly. Within a few hours, there were many changes. So we saw that the 2 genes were not regulated, but expressed in a completely unregulated way relative to each other, although they share the identical promoter. So we repeated this experiment, but we introduced a modification. You see these elements here, indicated by the arrow. These elements decrease the stability of the messenger RNA and the protein. So we took out these elements, and we hope that we make the messenger RNA and the protein more stable. And this is exactly what happened, indeed. So this was the original movie. I show it again because it's important to compare with the next. What you see here is a variation of the color in each cell. So the proportion of the two proteins changes very rapidly. Within the same cycle, you see that it can go from red to yellow to green and back. Because the half-life of the messengers is very short in terms of minutes, and the half-life of the protein is 2-3 hours maximum. So now, when we take out the destabilizing elements from the transgene, yes, this is just to show again that we eliminate the destabilizing elements. We still have the same heterogeneity in the population. So if you take a snapshot, you see exactly the same picture. You have cells with different colors, although they are all coming from the same initial cell. So diversity is total. And now let's have a look at the movie. Now, the first movie was on over a few hours, 10 hours if I remember correctly. This movie is over a week. So before we start the movie, what you can see, these cells are all from the same clone. So they're already diverse, as you can see. And the density is quite low because we'll make a movie over a week. But already you see that they have different colors. you see that the cells start to move around. They divide quite rapidly because, of course, over a week and we accelerate the movement very much. And they fill the space gradually. And what you can really, what is really strange here, what you can really see as a difference compared to the previous movie, that the cells transmit the level of fluorescence over divisions. So when the cell divides, the two daughter cells will have roughly the same level of both fluorescent proteins. As a result, we see patches of different colors established here. I can show again the movie because maybe it is interesting to see it again. So you can see, for example, this one is yellow. It will divide in yellow all the time. So a small subpopulation is constituted. The migration is not rapid enough to mix everybody completely. So by the end, what you can see is that the subclones, the subfamilies, have different colors. So this is just a snapshot from the middle. You can see that these cells are all coming from the same initial cell, already clonal at the beginning. So what we have here is that we have an initial heterogenization and also a kind of transmission of the fluorescence level over divisions. How this is possible? So what happens here exactly? If we try to, so if we try to analyze cell by cell, for example, and we try to analyze sublineages, what we can see is that this is the total fluorescence level during this, a single cell cycle. The two panels, left and right, show the same cell. So this is one cell, this is a second, and so on. And what you can see is that this is the maternal cell. The total fluorescence increases over the cell cycle. At the moment of division, it drops back to the initial level. Of course, because the total fluorescence so grew twice as big as it was at the beginning, contains two times more fluorescent proteins, and it drops back to the initial level at the moment of division, and the increase starts again. But the resolution of this figure is not good enough to see that these are 2 lines, these are 4 lines, these are 8 lines, because the number of cells increases. But you can also see that more or less the lines are superposed. So the divergence is not very big after cell division. You can also see that the two fluorescent proteins behave roughly in the same way. They both increase all over the cell cycle and drop back at the division. Longer exactly the same thing all the time. How can we interpret this observation? So it looks like that due to the higher stability, the proteins accumulate during the cell cycle without significant degradation. So the production rate is the double of the degradation rate. This is how the cell can double the amount of fluorescent protein in the cytoplasm during the cell cycle. Then the cell divides and the total fluorescence drops back to the initial level. It is because the total fluorescence is halved. So we have a correspondence between the cell growth and the rate of synthesis and minus degradation in this case. This makes that the average fluorescence level remains the same. So now we have, for example, another cell here. What you can see that the CFP behaves as I showed in the previous slide, but YFP protein is somehow decreases over generations. So what happened here is that after the division, or maybe during the second cell cycle, the transcriptional gene stopped, and there's no more production, and slowly the protein is diluted over divisions in the daughter cells of the initial cell. But at the same time, the CFP remains active, and so we have the same phenomena going on with the CFP. So what we have, we can interpret this in 2 different ways. Actually, they are not complementary. One is that we have a case of heredity here. Heredity in terms of phenotype. because the cells transmit the fluorescence level over divisions for many generations. We made the experiment, it takes more than 3 weeks to recover the whole distribution of the fluorescence from a single cell. So after 2 weeks, you still can see which is if the cells belong to the same subclone or not. So this can be seen a kind of heredity due to the stability of the components. This heredity works only if the production rate is high enough to double the amount of the protein in the cell, and if the degradation rate is longer, the half-life is much longer than the cell cycle. In this case, we have a half-life around 50 hours, which makes more than 2 cell cycles. So a protein synthesized here will survive normally to this stage, more or less. And we can see this here. And the second point here is that the protein stability or molecular stability in general, macromolecular stability in general, is not only an intrinsic property of the molecule. It reflects also the network interactions. Protein molecule has to meet a degrading enzyme to get degraded. So what we did when we put out the elements, destabilizing elements from the protein, what we did is we modified the network topology because he could not interact with some degrading enzymes anymore. So this is why his half-life became so long. Modifying the network topology means introducing change in the system's behavior, and we get a memory effect here, and we had no memory effect before because with the previous transgene structure, there was no memory at all in the system. So this can be interesting also if we think about heredity as a kind of network property. And if you look at the DNA molecule, it's really very stable. It is doubled exactly during each cell cycle, and the structure is maintained unchanged. So we can see DNA-related heredity or network property where the DNA and the meeting between DNA and the degrading enzymes is unlikely. We can simulate this with a simple model. What you can see here is the RNA fluctuations, which is really much higher than the protein fluctuation buffers RNA fluctuation, and you can see also the doubling and decreasing at each cell division. And if you take the normalized fluorescence, which corresponds to the total fluorescence divided by the cell volume, we have a stability over time. This is not the case if the molecule is unstable. You can see immediately that we decrease the number of molecules in the cells, and also we lose completely the cell cycle correlation, and also the average fluorescence will vary much more. And the autocorrelation, of course, decreases. So this is perfectly understandable in this way. Now, if you imagine that the cell contains plenty of different proteins and they all interact with each other, some interactions can be stabilizing because they protect the protein from degradation. And other interactions can be destabilizing because they degrade the protein. So this network topology will strongly influence the stability of the system over time. If you think about regulatory mechanism in the cell known by, the small RNAs, the siRNAs, miRNAs, and all these things, they all degrade messenger RNAs. So meeting between two RNAs is a part of the network topology. So then if you have an expression or non-expression of a gene expressing these siRNAs or miRNAs, can modify the network topology and can introduce a memory-like effect. And indeed, miRNAs and siRNAs are considered as a part of the epigenetic memory. So let's take another example. If we take, again, this landscape metaphor coming from Waddington, it is clear that the most important moment in the life of the cell is the bifurcation. So what happens, how the cell will behave here at the moment of choice, how it will go left or right. So we try to to examine this on a biological example. This is the hematopoietic system, the blood cell system. Blood cells are produced all over the life. The classical idea is that we have a multipotent stem cell at the beginning, and that multipotent stem cell can somehow launch the differentiation process by making a choice to going to myeloid or lymphoid progenitor state. And so this is the first bifurcation, and all the others are, all the other stages are this type of bifurcations. So we are interested in this. So what happens at the very beginning? This is an experimentally, it is possible to examine this stage experimentally, because we can isolate these cells, put them in culture, and to look at single cell level what happens here. So this is exactly the experiment we did. I'm not going to talk about the whole work. I just show one aspect of the work. All the other I just summarize here. So the process goes very quickly. It takes only 2 days, 2-3 days maybe. These cells are isolated. They are stimulated. And we observe 2 big changes during the first 48 hours. There is a first change. We analyzed each cell molecularly at the single cell level. So what we can see here, there's a huge change in gene expression during the first 24 hours. It goes from an undefined pattern to another undefined pattern. Actually, this pattern is undefined in the sense that each cell is different molecularly, and you cannot really classify them into discrete groups because there are no, they just form a cloud. This state is known as a multi-primed stage, which means that they express randomly basically every gene which can play a role in the lineage definition. So come back here. At this stage, after stimulation, these cells will express all the gene characteristics for every line in a random combination. So it looks like a totally stochastic state, but very rapidly, the stochastic state will boil down to 2 discrete patterns which correspond to the 2 progenitors. So it looks like that a choice is made somewhere here. So now, what we did with these cells, they put them under the microscope, and we looked how they looked at how they look like. And what we can see at 48 hours that there are 2 types of cells. So it fits perfectly with the fact that we see 2 different gene expression pattern classes, 2 classes of gene expression pattern. So one is a round shaped, and the second is a polarized. So what you can see here in blue is the cell nucleus. It's huge. It takes most of the space in the cell. And we see the cytoskeleton, the actin filaments in green. Clearly, these 2 cell forms, cell shapes, or cell morphologies are very different. It's easy to recognize them. And we knew from the literature already that other people observed the same thing, and they considered the 2 forms as 2 progenitor cell types, as I showed you before. So it looks like that the choice between the 2 pathways has already been done. So then what we did is that we made a time-lapse movie of this. So we put these cells under microscope. We managed to put 1 cell in each of these small wells. These are small wells, and cells are here. And we made the movie over 5 days. So the movie is a little bit long, and I can maybe accelerate it, but it's worth to have a look at least once. So what you can see is that at the beginning, the cells are very small. They're attached to the surface. You may have the impression that they want to break to freedom. And they will indeed do, but before they become bigger and bigger, so bigger and stronger, at one stage they will be able to he has already done this, so he is actively moving. This is a polarized cell, clearly, so he is able to move actively around. This is a movie which was done for over 5 days, 1 image per minute, roughly. So it's a little bit accelerated, of course. But you can see also the second cell also freed itself. And is also moving more or less actively around, but it's a little bit different already. What you can see is that the shape is closer to round than polarized, and they have this pseudopod coming out from the cell from time to time. What it has divided, I forgot to put the counter. The division occurred after 48 hours, roughly. What you can see that this cell is still alone, still running around, looking for something. At the same time, these cells start to divide very rapidly compared to this. And also what is interesting that they are mostly round shaped, but from time to time they go into polarized and they wander around the well. And then that became again round shaped, divided. And these are the two sister cells. Look up, both transformed again into polarized and try to run out, and then come back to the round shape and again to polarized and so on. So they regularly switch between the two states. It looks like they are looking for their identity. They don't know what to do. Should I stay or should I go? At the same time, the second guy also divided into two, so now he is even more active. They are really big now compared to the initial size. And they're running around all the time. They always polarized. They never go round, and except during the division. Here the second will divide. Now we have 4 polarized cells. On the left side we have almost 30 cells already, most of them round shaped. Sometimes some of them switch back to polarized and continue to move around, then switch back to round, and so on and so on. So it goes like this over 5 days. After 5 days we stop the movie. Actually, what happens is that we cannot go further with the system because the space is too small to house all these cells. So what you can, now it stops, so you can see that here every cell is polarized. Here most of the cells are round, but some of them are polarized, and they were able to switch forward and back all the time. So it looks, we interpreted this as a kind of dynamic process of fate choice, where you have an intermediate state, which is the switcher cells. Corresponds to the cells that hesitate between the two fates. So you have the round, they found their way rapidly. You have the polarized, they found their way rapidly. And you have a category of cells. If we count all the clones, we have several hundreds of clones now. If you count all the clones, roughly 1/3 of the clones contain hesitating cells. These hesitants correspond to the moment of the fate choice when the fate choice is not a unique event, but is a process which takes place sometimes several cell cycles. Sometimes it's very rapid, and then the cell can go to one or to other stable state. So we can make statistics on it, just for fun. There's also a small movie here. But what is really important that if you look at this diagram here, you will see that polarized cells, in 3 out of times of division produce polarized cells, but sometimes they go straight to round, sometimes they go to the dynamically switching state. These dynamically switching state cells, when they divide, also 3 times out of 4 give switching cells, but 1/4 of the time they go to round. And what is interesting, when if has cells with stable round phenotype, they will never come back to polarized. So this is a kind of sink here. The system goes to this sink, and most of the cells become round and stably round at the end of the experiment. We know the system will not stop here because we have so many different cell types in the blood, so they will go on to do other choices later, but we are interested only in this first choice. So again, this means that the choice is not a unique event. It's not just I go left or I go right. It's something which is dynamic, and it takes time to decide. So what we see here, when we try to isolate cells corresponding to this, that if you make a molecular analysis, that corresponds to a completely intermediate state between the two profiles. So these cells not only change morphology, they change also gene expression. So this morphological change reflects some kind of gene expression fluctuation within the cell. These are a couple of profiles to show you another interesting effect. So this is the maternal cell. As you can see, the first cycle lasted more than 60 hours. It was polarized all the time. Can produce two daughter cells. They polarized at the beginning and they start to switch, and you can see that profile is very similar. The time profile of the switching dynamics is very similar. So there's a kind of memory again here, which makes that the two daughter cells behave in a very similar way, and we see this very frequently. Also, the length of the cell cycle is very similar in the sister cells. These divide into two daughter cells, and this also two daughter cells. Here again, you can see a conservation of the switching pattern, which is extremely similar in the two daughter cells. Here it is a kind of, you can see a little difference. This is another one, very simple. It was polarized from the beginning and remained polarized all the time, except during divisions. A third one, I don't know what is this. A third one, you see this maternal cell divided after 40, 50 hours roughly, and it was mostly round-shaped with infrequent switches, produced two daughter cells who started to switch and then stabilized in the round shape, and the granddaughter cells which mostly round shape. So here the stable round conformation morphology is acquired. So again, we see here variation and memory. Variation and memory in the same cell line. So it shows how these two things are really complementary, and one is somehow, the two things go together all the time. Small variation means strong memory, high variation means lack of memory. I show another example because it is also very interesting, somehow different. It shows that the cells, due to their internal variation, can adapt basically to any condition, even if they meet some completely unforeseen circumstances, they can adapt easily to them. In this case, for example, these authors cultured the cell line in microgravity. So they put the cells in a machine which made random movements which ruled out the effect of gravitation. And I talked to the authors, and they repeated the experiment also in sending cells to the space station. And it turned out that initial homogeneous population will split into two morphologies. In the absence of gravitation. Here you can see the two morphologies, so different cell skeleton proteins are labeled with these antibodies, so you can see that the two morphologies are different, and they are different from the initial morphology. So this is a summarizing figure here. So what you can see is that under the effect of, or in the absence of the effect of gravitation, they go out from the stable state and they split into two pathways, and they acquire two stable states. This point corresponds to a moment of hesitation, so they can see these hesitating intermediate states. And what is interesting that if they put back, if they stop the experiment, that they put back in the normal gravitational conditions, the cells will come back to the initial state. So this example shows that first of all, that this choice is certainly not genetically programmed, as we believe. So there is no regulation here, because no cell in the world has ever met the lack of gravitation before. So there cannot be evolved mechanism to regulate how to adapt to the microgravity in a living cell. So this is a perfectly unforeseen and unscheduled condition that cannot call for an already developed mechanism to respond to it. This shows exactly this nature of the systems based on stochastic elementary events to be able to adapt to any condition. So this is the summary figure showing that what they see here is that before you may have small stochastic variation in the cell, and then at the moment of choice you have a huge variation, you have a huge disorder state. So you go from one order to another, you have to make a disorder in between. This is what happens here, and then they go to one state to another. So this brings us to the question of how the change is initiated. So, okay, you have a stable state here, you have two stable states here, but how would this change is initiated? What is the trigger? In the deterministic view, this is what we used to say, that there are master genes, a master gene is switched on, and it will activate a pathway, and it will lead to a phenotype change. So this is the traditional view. If you remember yesterday, I talked about the preformationist theory, which is a kind of deterministic, strongly deterministic theory. This type of explanation faces the same problem. So who regulates the regulator? There's also a regulator of the regulator and the regulator of the regulator of the regulator. So there are only 20,000 genes in the genome, so you cannot go very far with this logic. So there has to be something else. This means also that if we rule out the master gene, so we have only the genes which have to be expressed differently after the choice, fate choice. We have to see how things happen in between. So what will trigger the change? One stable state will change to another one. The deterministic view can be represented in this way. So actually, what is always difficult to explain is the beginning of a process, how it starts, how it will go out from the stable state. In this deterministic view, the causality is linear. This is what we explained. And when we are talking about stochasticity, and if you, what I said at the beginning, that we inverse the frame and we put variability as the ground state, and we need to explain stability, then we don't need to explain what will trigger the change, because the variability is there. It's enough to not to stabilize. Enough. Not to stabilize. So we are really dealing with the question of stabilization and not with the question of how the change is initiated. So how a gene switches on or off? Because we all agree that a differentiated cell will express genes differently than a non-differentiated cell. So this is a simple cartoon you can find in the textbooks to explain how a gene is expressed. So the blue line is the DNA. These small cylinders correspond to the nucleosomes, to the chromatin. And what you see here is the promoter. I showed already a figure like this yesterday with all the proteins binding to the regulatory sequences and initiating transcription. So this is a gene on state. And this can only be done if the chromatin is decondensed. I mean that, you know, the DNA is never free in the cell. On the cell nucleus is always associated with proteins, forms the so-called nucleosomes that are represented by the cylinder, as I said. And even these cylinders can associate into a more condensed structure, which is the, we used to call this heterochromatin or condensed chromatin. So when the DNA is in this stage, no transcription is possible because the DNA is not accessible to these proteins. So to get access to the DNA, this structure has to be dissociated first and leave the DNA sufficiently accessible to initiate this process. So then the transition between the condensed and decondensed chromatin is the key of the question. The problem is that if DNA is not accessible, then how activators can find the gene to activate? It is clearly not possible because DNA is not accessible. So imagine that you go in a library and you want to find your favorite book, but there is no catalog and there is nothing written on the front page of the book. So how you can find the book you are looking for? Different strategies are possible. You start for the first and the second and third, and you go all over the books, or you can make a random search, and probably if you make a random search, then you have a chance to, if you are lucky, you will find it quickly. If you are unlucky, you will not find it before a long time. So this, what happens here also, the genes are activated randomly, and then if the good gene is activated, it will be stabilized, and the gene which is useless for in that environment will be switched off again. The key is here, how to open the chromatin and how to close it again. For this, we have a system, biochemical system, which is actually this is the other way people used to define epigenetics. Chromatin components, these are nucleosomes again, schematically, carry covalent modifications, and those covalent modifications influence the stability of the structure. Some modifications destabilize and dissociate easily the nucleosome from the DNA, while other modifications are acting on the opposite way. They're stabilizing the chromatin and they make it more compact. So there are different type of, many different modifications like this. ADP ribosylation, methylation, acetylation, ubiquitination. So all this is well known in biochemistry. We know the enzymes that catalyze the on and the off reactions because each of these modifications is reversible. It can go on and off with a very high frequency actually, because all the enzymes are present all the time in the nucleus. So actually the condensed or decondensed state of the chromatin is a steady state, the dynamic steady state, which corresponds to the balance between the on and off reactions. The reactions cooperate between each other. So one reaction which is destabilizing, one type of modification which is destabilizing will favor, promote another reaction which is destabilizing and the opposite. So it's a typical situation when you have two stable states. Schematically, this is the list of type of modifications you may have. The details are not really important here. What is important that they are reversible. Each modification is reversible, and now we know that it is also involved in the transmission of the chromatin states over cell division. So it's also a kind of memory system working in the cell, depending on the frequency of the changes, of course. Let's not to talk about this in much detail. Have a look at this part of the figure. So if you look at the half-life of these complexes, what you will see is that the transcription factor residence time measures in seconds, but the nucleosome residence time can be measured in up to hours. So it's really a stable structure compared to simple interactions with the polymerase and promoter, for example, or transcription factor with the promoter, which has a residence time which is very short compared to nucleosomes. Nucleosomes are really stable structures. In terms of evolution, nucleosomes are highly conserved. Each eukaryote has basically the same type of nucleosomes. You can replace the yeast's histone with a mammalian histone and it will work perfectly. So the stability is the key component. So people try to examine the stability in many different ways, of course, and I cite only this study because I found it's really, really compelling. This is a theoretical modeling of the chromatin, a simplified model. The model is based on the idea that the chromatin may have three states. The M corresponds to the repressed chromatin, the A corresponds to the active chromatin, and the U corresponds to the situation in between. So when it has no modifications for stabilization or destabilization. Since it is reversible, from going to A to M, it has to pass through U, of course. And the enzymes that will initiate this transition by putting a modification which corresponds to activation or inactivation, also removing the activating or inactivating modification from the nucleosome. So you have these on-off reactions simplified as a single modification. Then you have also the two types of chromatins, so a stretch of nucleosomes, and you see that there is a cooperative effect. So if one nucleosome carries a modification of one type, it will increase the probability of the same type of modification around the neighboring nucleosomes. It's the same for the active chromatin. So there is a cooperativity, it's very important. And now what we can see, what we can guess that if you launch the system from zero, let's say every nucleosome is unmodified, it will go either to the silenced or to the active state. So to get transition between the two, we need some noise. So they introduced also the possibility of noise. Sometimes some modification occur randomly. So not every modification is cooperative. Some of them are absolutely random. And this is the result of the simulation. So if the noise level is very low, you will sometimes have a switch from active to inactive. Actually, this is from inactive to active, I think. Doesn't matter. If you increase the noise level, you will get more noise and more frequent switches between. If you further increase the noise level, you have really a lot of switches, and it's becoming disorganized, completely disorganized if you pass the noise level to very high. Nothing surprising. Basically, this model just confirms what we already know about the bistable systems, and we have discussed already many times this week. This is really a classical bistable system without any surprise. What is original in this model is that this is a clear demonstration how noise can initiate a switch in the chromatin state and open the chromatin and make gene expression possible. So we understand what kind of modification can stabilize and destabilize. This is a whole literature about the biochemistry of the chromatin. So where this noise comes from. So what is the noise here? This is just a representation how a stable system can work. So it turned out that when people started to accumulate knowledge about the chromatin modification, everybody focused on the enzymes first. As usual in biology, we are interested in enzymes and not what the enzymes do exactly. And only later they paid attention to the fact that each of these enzymes, every modification, every chromatin modification, without exception, uses a small molecular substrate, a key molecule of the energy metabolism. So intermediates of the Krebs cycle or glycolysis or ATP, the end product of the oxidation, are used as substrates to modify the chromatin. So this means that there is a mechanistic link between the energy system of the cell and the chromatin state. And this is experimentally demonstrated now. So you can modify things in the metabolism and you can get a chromatin change very easily. You can also initiate cell differentiation or block cell differentiation if you act only on the metabolism of the cell. So then we are coming to the final point. Metabolism, you heard earlier, it's a complex network of reactions. Again, we are dealing with a network. Network means number of vertices and links between them. If you look at a metabolic network, it's very different compared to a gene expression network, for example, because you have vertices which are the small molecules and not the enzymes. And in a gene expression network, we consider vertices as the proteins coded by the genes, or the RNAs maybe coded by the genes. Here, proteins are the links between the vertices because they catalyze the transformation. There are also some other differences, of course, but what is really interesting that here a network state corresponds to the concentration of each element, even without changing the protein composition, the enzyme availability. The same network having exactly the same enzymes at the same level in the same cell may have two alternative states. So I take here just a single example of a metabolic flux simulation where in a ground state of the cell, you may have a network optimizing growth, biosynthesis, and the same network, an alternative stable state of the same network is the ATP maximization. So the two major sources of energy, which is the glucose and the glutamine here, contribute differently and to different extent to each part of the metabolic network, to each branch of this network, they can contribute differently. And they can reorganize spontaneously between the two states. So why this is important? This is important because depending on the two different states of the network, you have the concentration of the key molecules that are different in the cell. Sunil mentioned acetyl-CoA as a key molecule for the yeast oscillation. Acetyl-CoA has a central role in every process in metabolism and also a substrate for chromatin modification. NAD, also Sunil mentioned it, also a substrate for chromatin modification. So when the metabolic network switches from one state to another, this will involve change in the small molecular component concentration, and that can induce a change in the chromatin state. And since the chromatin states will change, new gene expression change can occur also. So very rapidly, this is a logic showing how we can go from metabolism to chromatin change and gene expression change. So, and what is really noticeable in the metabolism, that small molecular components are not limited by the copy number. So if we have a small copy number for proteins and RNAs, metabolites are always represented by a huge concentration in the cell. So they are at a millimolar concentration in the cell, which represents a statistically significant amount of statistically significant number of molecules in the cell. So we are not limited by the stochastic effect when we are talking about metabolic effect, because they are buffered by the high concentration of the components. So let's go for a summary. The question was how cells function reliably with such an inherent variability. Okay, there was an averaging effect, okay, and also intrinsic and extrinsic constraints. So intrinsic constraints are generated by the system itself. I use the term system instead of using the term cell because what is intrinsic at one level of organization can be extrinsic on the other level of organization, and this holds for the extrinsic constraints also. For example, molecular variation in a single cell are intrinsic to the cell. They create heterogeneity of the cell phenotypes, but heterogeneity of cell phenotypes will be intrinsic for the population, but extrinsic for this at the cell level. To try to explain the difference between these so intrinsic and extrinsic is a relative, two relative concepts. We have to keep this in mind, and to explain it in a simple way. So this is a regulated system with noise. Regulated system with noise, you have an average behavior of each element, and you have a small noise around this average behavior. if they are beginners, the noise is bigger, and if you are professionals, the noise is smaller. But now, if we look at a system which is constrained by the extrinsic constraints, you also have a ordered movement in the same direction, but that movement is not determined by the regulated behavior of each individual. It is constrained by extrinsic constraints. And now, if we have a look at a system where the elements have intrinsic constraints, that can also generate a kind of order. For example, flocking. Here, there are no extrinsic constraints because birds can go anywhere, but still they have a regulated movement because they are behaving according to their own intrinsic constraints. The intrinsic constraints are the simple rules that drive their movement relative to each other. So now if we put together these two, or you can even add a little bit of this, we can get something which will behave in a very nice ordered way, and as an observer, or by observing it from the outside, we may have the illusion that everything is completely regulated. So, extrinsic constraints generated by the environment, and as I said, that what is environment at one level can be intrinsic at the other level. and what is important also that usually when you consider the intrinsic constraints, it can define a state space with many different possibilities, but not each possibility is accessed by the system. So actually this slide should have been before. But when we consider the extrinsic constraints, we can also classify them into different classes, like all this. The basic idea is what I said, that you may have a system which is completely disordered at the beginning, and that if you put constraints on it, either from the outside or from the inside of the system, you can get a perfectly ordered behavior, and this is what I think happens in biology. So, thank you. If you have questions, it is now, but if you have, when you finish the questions, please stay for a moment, because there is a small announcement to do after, okay?
So am I correct in understanding that you're ascribing a central role to metabolic state of a cell?
Yes.
In the whole model?
Yes. If we look at the time scale of changes, for example, what we see that the cell differentiation is something which takes hours and days, as we could see. If you look at the underlying gene expression changes, they take hours, best minutes. And if you look at the metabolic changes, they can take less than a minute. So you have an initial change which can come very rapidly, and it can trigger the second, which takes a longer time, and it trigger the third, which will take even longer time. And then we can get the evolutionary time scale. Changing DNA takes years, centuries, or thousands and millions of years. So the system of the memory relies on the stability of its components, and depending on which component is considered, you may have a time scale which is longer or shorter.
The question which follows is, how closely or how narrowly do you think cells can be focused to come to the same metabolic state? And if you do that, what do you think might happen in a population?
So the cells are an open thermodynamic systems, so they need to dissipate energy all the time. So the main focus of a cell, really the most important thing of the cell is to eat and digest, because this is the only way to survive. So this has to be the major drive in everything which happens in the cell. differentiation is a way to find the way to get more efficiently the necessary amount of energy. So what happens is that if all resources are unlimited and available, the cell needs nothing to do. You can just take up the food and produce the necessary energy, and that's all. But this is only this happens only in the laboratory, actually. So this is why when the resources are limited, a kind of heterogeneity will establish, and kind of cooperativity between the cell, a division of labor between the cell will be established. Not necessarily cooperative, but it will become a cooperative system very rapidly. So the most efficient way to use the whole energies, the food supply, is to specialize into different parts and different types of molecules and create a kind of network, metabolic network between the cells. This is what we see in the case of the Sunil presented to us in the yeast, for example, with the oscillations. Then the cells pass from one metabolic state to another depending on the amount of oxygen which is available. But we can see this type of cooperation in every multicellular organism. The best example I think about is in the nervous system. You have astrocytes and neurons. Astrocytes consume sugar, glucose. They transform into lactate. Lactate is rejected from the cell, and lactate is taken up by the neurons, and it will oxidize it completely into CO2 and water. The result is that neurons get a lot of ATP because the oxidation will provide them something like 34 molecules of ATP per 2 molecules of lactate. And so they can use this ATP to perform all the tasks they do, membrane potential essentially, which is highly energy consuming. At the same time, astrocytes, they get less ATP, but they can proliferate back because they conserve some carbon resources that can be used for biosynthesis. So this is a typical case of metabolic cooperation, which leads to two different cell types. And those cell types, the existence of those two cell types is absolutely necessary for the functioning of the system as the nervous.
Yeah, but my question was somewhat different, which is if you can buffer a population of cells, so to speak, to be in the same metabolic state, would you predict that you would not get differentiation, or that differentiation won't be marked?
I think that a lack of differentiation is only possible if the amount of the resources is unlimited. Under constrained condition, there will always some differentiation. Simply, I cannot imagine how cells can stay in the same state for a long time if they compete for the same resources. So some cells will get more resources and will become stronger. The others will get weaker, and I can imagine that the outcome of this competition is the outgrowth of some cell clones over the others. But in any case, there will be difference between them. However, if you have unlimited resources, then the cells don't need to cooperate. They can survive without cooperation. So they can continue to grow almost exponentially, and no cooperation is needed. This is what we see in the case of the cancer. Cancer cells, they grow rapidly and they don't cooperate. And why? Because they have unlimited resources in the organism. The best way to prevent cancer is to eat hypocaloric. It's well known that to prevent cancer, when it is established, that's different. But to prevent cancer, you need to eat hypocaloric and make exercise to increase the oxygen concentration in your organism. It's much better than any kind of other treatment. We also see that in the embryo, in the developing embryo, differentiation starts at the regions which lie further possible from the resources. So the blood vessels, for example, because blood brings the nutriments and the oxygen, and there is a very sharp gradient which is established because of the slow diffusion of these molecules out of the capillaries. And those cells that lie at the third or fourth layer of cells, so let's say 50 microns from a capillary, receive much less oxygen and much less nutrients. Those cells are the first to differentiate in the embryo. Those who have direct access to the resources, they become differentiated much later than this access starts to decrease.
What about selection? Because people like to talk a lot now, even in the differentiation, like lymphocyte literature about selection. Yeah. But as far as I can tell, except for now the cancer, there's much less of a role in it, in your point of view.
Mm-hmm. Well, selection is another way to define constraint. So it's a kind of constraint. Yes, but the processes initiated by the metabolism are not shifting so rapidly. So of course you can have fluctuations in the metabolism very rapidly, but once they trigger a change and they push the system into an alternative state, then that alternative state will remain stable for a longer time. Just because that, for example, you can also change the concentration of the enzymes, so you can open new pathways in the metabolism and close some pathways in the metabolism. So the initial metabolic change occurs without gene expression change, but that can trigger change in the gene expression and that can rewire the metabolic network also. Okay? The term selection of course, can be used in this case, but we have to consider selection as a kind of constraint. That constraint is generated by the system itself also, in some cases, and in some cases it comes from the outside. In this way, there is a guy who wrote a book about this, and he considers development. His name is Kupiec. He published his book in English also, and he has a theory where he considers ontogenesis as a process homologous to evolution, biological evolution. So he's saying that there's only one type of theory valid for the whole, for every level of organization, from cells to big populations, is the theory of selection, transmission with variation plus selection, where selection means all these constraints together.
I just wanted to add a comment to what everything Andreas has said and pertaining to Vidya's question. I think it's important to remember that metabolism is always in a non-equilibrium state. And if you think of it as something that can never be in an equilibrium state, it is easier to think of how you can get differences, because in any metabolic reaction, there is a flow which will result in the next thing also flowing. And with very slight differences in the trajectory of the flow, which is what largely Andreas talked about here, you can get multiple outcomes very, very easily. So there is this constant entropic, it's an unstable system. So you will always, regardless of how you start, even if every cell starts the same, you will have this outcome, which is differentiation or different states forming, because you're starting from a system that will remain in or continue in non-equilibrium. The only way you reduce that is, like Andreas said, when you have unlimited supply of something. That's the only way you can shift the equilibrium, shift the system closer to some kind of a steady state where the supply is dominating and so you don't get differences coming. But in every other system, which is what is common, you will have multiple outcomes. And I think it was very, very nicely explained in all the examples that Andreas had. So I just wanted to add a comment to this.
Thank you. I fully agree, and I also go in this, further in the thinking, since we are talking about evolution also. Actually, the basic design of the metabolic network is the thing which is the most conserved in the evolution. So you have the same glycolysis TCA cycle in all living beings on the Earth. It is much better conserved than the gene code, for example, the DNA code, which is different between prokaryotes and eukaryotes. So prokaryotes and eukaryotes all have the same basic network design for that makes, let's say, suggests that maybe metabolism was there before the genetic code.
So according to this view, the metabolic process can be seen as a kind of a sensor system. And then if the environment is changing, then it will sense it very quickly because all these fluxes that are not in steady state. And then what it will trigger the gene networks to make the good enzymes and then to adapt. That's the way you think all it's interconnected?
Yes, you're right. So basically what's happening at Sunil said also that metabolism can react very rapidly and can react continuously because of the high number of molecules involved. And that continuous fluctuation of the metabolism can just buffer the environmental effects and maintain the system on the same track all the time. But if the conditions change more, that then will change even higher fluctuation in the metabolism, and that high fluctuation will trigger other changes like gene expression changes. So in this way, metabolism plays a role of variation generator. So this is an amplitude modulated system and not a frequency modulated system. That environmental changes will act on the amplitude of the fluctuations, and those fluctuations become much bigger than that will generate additional changes to readapt the system to the changing environment.
But then what is the system that will stabilize the gene expression? Because you said, okay, gene expression is very stochastic.
The establishment of the metabolic flow.
The metabolic flow will stabilize the gene expression?
Yes, because there's details I had no time to explain, of course. So my hypothesis is that, and it fits quite well to some data available, that then metabolism is completely deregulated. What will fluctuate mostly, those are metabolites that open the chromatin. Okay? And when the flow becomes more close to equilibrium, the metabolites those metabolites will decrease, and the metabolites that will increase are those who stabilize the chromatin. Okay, so there's a kind of play between these two types of modifications. So when system comes back closer to a stable metabolic state, then it will stabilize the existing gene expression pattern.
Okay, and then you lock.
Chromatin plays a role of repressor. And contrary to the lack operon repressor, it is based on the cooperation of many molecules, because you have nucleosomes, you have histones in nucleosomes, and you have many nucleosomes, not just one repressor in per promoter. So this is a very powerful repressive system that represses gene expression. Putting repression on the chromatin will conserve the gene expression pattern at the state at the moment this repression starts. So this is how metabolism can integrate without acting specifically on that or that gene, can generate unstable situation in the genome. That unstable situation will allow the re-expression of many genes, totally stochastically. This is what we see, and in the publication you did also, we see this, and many others. And then the situation comes back to normal. Switch off starts, many genes will be switched off, and only genes which are really necessary for the survival of the cell will remain active. Because if they switched off, that will create again a stress which will push back the system in this random searching state. And if you compare the timescales, you will see that it is compatible with the timescale, because molecular events are so rapid compared to the cellular event that you have many attempts to find the good configuration. So it is not as, this is how we can see cells fluctuating for several generations, because they are looking for what to do. And once they find the correct combination, they can stabilize their phenotype.
No more question? Okay, so let's thank the speaker again.